Okay. 2, 4, 6, 8, 10, 12, 13, 15, 16, missing 1. Is that right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 16.
This is Lucas stuff.
Yes, Luca. Okay. Is this yours?
No, it says okay.
Okay, so we're going to talk about fluorescence by phytoplankton, pigments, and sedum. And then I'm going to tell you my bias is towards phytoplankton because there's a lot more, well, there's physiology in there. More people measure fluorescence. And sedum fluorescence is complicated, and I'll tell you as much as I know. And I will tell you when I've reached my limit of where I am at. Okay. So I'm going to start with the take-home messages because I'm not ever getting through my lectures. So I just want to make sure that we're all like on the same page of what. So fluorescence is the property of a molecule to re-emit absorbed light energy at a longer wavelength or a lower energy. Okay. Only certain molecules can fluoresce. So chlorophyll A can fluoresce and some organic molecules. And it tends to be a unique fingerprint to that molecule. So for example, chlorophyll fluoresces, and that fluorescent signature is unique to chlorophyll. So therefore we can use it as that unique identifier, which is really nice. It's really easy to measure. It's really difficult to interpret. And so if Kurt hasn't talked about the conservation of misery yet, let me introduce that concept. If something is easy to measure, it's usually pretty tough to interpret. If it's easy to interpret, it's probably really hard to measure. So sedum has a really complex composition, and so it's really difficult to sort of discern what it is you're looking at when you make a sedum measurement, although you can just stick a sensor in the water and get a number. Chlorophyll is a proxy for biomass, phytoplankton biomass. It can be the proxy for carbon. It can be a proxy for photosynthesis. It can be really a proxy for also nothing because it's so complicated to interpret. So everybody measures it, but you know, there's a lot of things that we have to think about. Okay, so this is basically what we're going to try to talk about today. What's fluorescence? Who does it? What is the physics of fluorescence? The different proxies that we use. I'm going to talk about in vitro, which means in a test tube, right? Something that's extracted, say a chlorophyll molecule in an extract of methanol or acetone. In vivo means it's the fluorescence in the living cell. And in situ means it's actually the measurement that we make in the water column. So it's a bulk measurement of who knows what is in there. So I'll talk about those 3 terms. Talk about calibration. The reason we're talking about calibration today is because this is what you guys are going to do in the lab. We've never really done a fluorometer calibration in this class, and I will tell you that most of the people that contact me, former students who have taken this class, but a couple years down the line, they call me up and they're like, yeah, so I just got a new fluorometer, how do I calibrate it? So I'm like, I'm just going to kill 2 birds with 1 stone. You guys are going to do some calibrations. And it's not going to be the perfect calibration because we're not going to, you know, do as many dilutions as I typically do, and the conditions in the lab are not perfect, but we're going to at least go through the motions. And so I'll show you a little bit about how to do that, but then basically you'll be on your own for the afternoon. And then also, we may not get to this today, but we'll talk about it through the next couple of weeks. Given the sources of variability that lead to variations in fluorescence, what can we use fluorescence for to learn more about the environment and more about the phytoplankton and more about who they are, their physiology, and their environmental conditions? Okay, sound good? You're scowling.
Oh, sorry,
It's okay.
I'm not sure what you're trying to get at, but I'm sure I'll know by the end of the lecture.
Yes, source, yes, you will. Okay, so you did this probably in the first lab, right? Where you take something in under visible light, this extract of spinach looks green, you put it under an excitation light and you can see the fluorescence glowing red. It's really beautiful. If you look at a cell under a light micrograph, You see the chloroplasts. If you look at phytoplankton, and unfortunately it's not the same image, it's really hard to find replicate images with an epifluorescence microscope. If you have one, please send it to me so I can put it in my slideshow. You can see the fluorescence by the, this is ectomorpha, and you can see the individual chloroplasts fluorescing there, which is really beautiful. Okay, so that's who does it, phytoplankton are fluorescing. I'll show you the Sedum later. Okay, so here's where we go. You guys each have an energy level diagram, and we've talked about the energy levels for absorption, and I want to sort of have you think about what that means for fluorescence, okay? So we have energy on the y-axis. We have the ground's electronic state here. You all have that. I want you to sketch the electronic energy levels for the two absorption peaks of chlorophyll A. Just the electronic levels, not vibrational or rotational, okay? And I want you to use color-coded arrows, blue and red. And I want you to draw the spectral representation of those two electronic absorptions. So go ahead and do that. You're gonna be like 2 or 3 people per graph. So see what you remember. Oh, yeah. Yeah, so what I've given you is, I've given you this. You have to sketch this yourself. I didn't give you an axis. You might have to do your own axis here. This is wavelength. This is just the ground state energy level. We're just doing absorption, just absorption. So you're not doing fluorescence yet, just absorption. Yeah, you're just sketching the absorption peaks for chlorophyll. Good. So, and then draw yourself a spectrum underneath. It's not working. Well, she said, okay. Okay, you feel good? All right, okay, so does everybody have the first electronic state? And the second electronic state, that's at a higher energy level, right? Yeah, that looks good. And we have absorption in the blue, right? Because this amount of energy from the ground state to the second excited state is more energy. Blue wavelengths are more energy than red wavelengths, right? Awesome, good job. And that's associated with 2 discrete absorption, narrow band, right? Only associated with the electronic transition. Awesome, that looks great. So you guys have full on spectra right there. You're not there yet. It takes the vibrational and the rotational levels to fill in those, right? Excellent, okay. So now, we're asking the question, so what's going to happen to this absorbed energy? We have these molecules, they've absorbed this much energy, this much energy. And so this is where, when Ken is talking about the lifetimes, that absorption going into the molecules, it's holding on to it for a nanosecond or a picosecond, which is really long on the time scales of energy dissipation. So what happens is, for phytoplankton, who are trying to use this absorption for photosynthesis, it turns out they really only need the energy equivalent of a red photon to perform photosynthesis. So this excess absorption in the blue is just going to waste. And so that amount of energy gets lost as heat. And so we show that energy loss is, you know, infrared heat. So it's a squiggly black arrow. Right? So very quickly, the molecule will de-excite down to the first excited state. And so you can imagine that's a lot of heat that the phytoplankton are going to have to get rid of, because heat denatures protein. So this is not something that they love to do. But unfortunately, for phytoplankton, most of their light is in the blue wavelengths in the ocean. So they have to deal with it. Okay. So what about the energy from S1? So ultimately, that's energy that's going to go to photosynthesis. But it doesn't all go to photosynthesis, right? I mean, that's primarily where it's going, but if there's, and I'll show you there's reasons why there will be excess energy that they have that, you know, needs to be dissipated. So the energy from here to here, what happens to it?
It re-emits.
It re-emits, right? And so that's where we get fluorescence emission. Fluorescence emission is coming from the first excited state, the first electronic state. And so to represent this on the energy diagram, how might we represent that with an arrow?
Red arrow going.
Yeah, red arrow going down, right? And so it looks just like this arrow. How much energy does that emission represent?
The same amount as the first going up, but
Exactly.
in a different direction.
Yes, it will be emitting away from the molecule, but it's exactly the same amount of energy, right? So where is it going to show up in the spectrum? Exactly at that wavelength, right? And so for those of you who know anything about fluorescence, you're immediately thinking like, I thought it was shifted to the red. We'll talk about that. This is the first representation of fluorescence. This is exactly what we would see. It absorbed, it re-emitted, it would re-emit, there would be no change in energy, it would be exactly the same wavelength. Okay, it's not, but we'll see why in a sec. Okay, so now what I want you to do is add 3 vibrational levels to each of your electronic levels. Okay, and you can do that with your black pens. Then I want you to add absorption lines going from the ground state to each of the vibrational levels of the electronic states. So these are the vibrational harmonics. And then add that to the spectrum. Okay, so go ahead and do that.
Three to each. Three.
Yeah. So remember what the vibrational levels represent, right? They're representing the additional energy, kinetic energy.
They're all a bunch of
So are these vibrations associated?
yes, right, but yeah, this is so the vibrational are
I'm not gonna have you do rotational, so you don't have to put the rotational in. You'll get two messages, just the vibrational. And there will be 3 vibrationals on each of the electronic states. So think about the ground state, and then think about vibration means there's additional energy to vibration. So if there's additional energy, it is in higher ability in the electronic state.
You can excite me.
Oh,
I think there are these. Well, no, here, oh. So they also are
Oh, yeah. No, I think. Yeah.
So what I'm looking at is like, so then I guess.
So remember.
This is not blue anymore.
Yeah, so it's gonna be like this. It's, remember, it's like.
Yeah.
But that's associated with the probability.
This is just arbitrary.
If you want to simplify, you can draw from the ground state. If you want to do from the ground state of the S0, I mean, or you can leave it the way you have it. It's more complicated than I had it, but you are happy to do that. See, it's so much easier when I just do it on the board, right? Now, this is why we're doing this, because it'll make it. So you imagine you've got your ground state and you absorb to that second electronic state. Now, what if the molecule has a vibrational energy to be a little bit higher? So you'll absorb a little bit more from the ground state, right? So it'll be, yes. Okay.
Are we?
So where are they under electronic? Higher energy. Higher energy.
There's the.
It's higher energy level.
I don't think it matters if you draw over under because
But I was like, as far as the height.
Oh, we don't know about their height yet. We know nothing about their height yet. I'm giving you no information about that. Yeah. Okay.
Well, you did give us information about the height in the previous lecture.
I did,
You said the
but
probability.
I haven't given it to you yet. I haven't told you what the probability is. All right, do you feel like you have, I think some, there, yep. Okay. All right, so let's reason through this. So we have the ground state and we have 3 vibrational levels. We have that for S1 and we also have it for S0. Okay. So in the simplest case, if we started in the ground state of the lowest vibrational level of this ground state, and some of you actually put in absorption from the higher vibrational levels, what does that represent? So I put them all from the ground state to try and keep it simple, which meant that you go from the lowest vibrational level of the ground state to various vibrational levels in the electronic state, highest electronic state. So this was our original one. This is more energy because you're at a higher vibration, right? And more and more. So where are those going to show up on this spectrum? To the bluer end or the higher energy, right? So when we get to this one, I don't know why I drew them in another order. It's just because I ran out of space and I didn't want to move my arrows. Again, this was our from the ground state of the lowest vibration of the ground state to the first excited state, and then to the higher vibrational levels, more energy. They're going to show up on the spectrum. More energy, which direction? To the higher energy end, right? So I've told you nothing about the probabilities, so we know nothing about the relative magnitudes yet. Those are the vibrational harmonics of absorption. Now, if the molecule was not cold, we could have started from one of the vibrational levels of the lowest energy level as well, and you could put those in as well. And if that were the case, and we went from here to here, that's the least amount of energy. So that would give you sort of some side peaks this way, right? Okay. All right, so now what happens is we get heat loss from the vibrational levels to the lowest electronic state very quickly. So we lose this energy from here down to here very quickly. And we lose the energy from here to here very quickly. And I've already told you that once we get to the second level, we lose this. So we end up having heat radiation and heat radiation. And then all of this energy goes down here. So suddenly we end up having a lot of energy at this first electronic state. And all that energy is going to, or the fraction is going to decay as fluorescence, right? All right.
Wait. The heat energy is the fluorescence?
The heat energy is just the loss from the vibrational states to the ground state. And in fact, if you look at what that energy equivalent is, it's a near IR photon equivalent. It's a heat. Heat is in the near IR. So it's essentially like you're radiating IR. Yeah.
I thought he was like 12 to 20 microns.
Yeah.
Like,
Yeah. Okay, so the fluorescence from here to these vibrational levels now. So we just had this one, which showed up exactly in line with our absorption. But now, if we go to the first, second, and third vibrational level of the ground state, yeah, what's that going to look like on your emission spectrum? It's going to be longer wavelengths, less energy, and it's going to be quantized by the vibrational level, right? So it's also going to show up as, so that's that, da-da-da. It's going to look like this. There will be harmonics associated with fluorescence.
This is just happening for a small subset of photons that are not also in photosynthesis, right?
Yes, yes. So if it's an extract, there is no photosynthesis. So for a molecule in extract, it's happening a lot. For a molecule that's in the cell, there's a quantum efficiency depending upon all of the physiology of the cell, but enough that we can measure it. Yeah.
Is there again heat loss between the states?
Yes, then there would be heat loss there too. Yes, I didn't put that in, but you're right. Yeah. Okay. So the probability of occurrence of any one of these states depends upon the internuclear distances in the molecule. And it turns out that there are some that are more likely than others. And that's what gives us these magnitudes. And it turns out if it's a solid line, it's the most likely to occur. If it's the dashed line, it's the next most likely to occur, the third most likely to occur, and the least likely to occur. And if it's really lightly dotted, like for example, this one, it doesn't occur very often at all. You can almost ignore it. So what we find is that the primary peak is at 440, and you have these smaller harmonics into the UV. In the red absorption, we have a peak at 676 with harmonics going towards the green. And the fluorescence, you see the main fluorescence and a side harmonic peak But what's interesting is that with this part of the spectrum, they end up being almost a mirror image of each other. And it's because of this quantization of fluorescence and quantization of absorption. Yes.
So the most likely in the red does not go directly to the energy state. Is that
No.
like the precise amount of energy they need to photosynthesize?
Yes. Yes. Yeah. Except for when we get to talk about reaction centers, you'll see that there's actually two different wavelengths that they need, 680 and 700. And the molecules are tweaked. Their bonds are tweaked so that they maximize that. Yeah. So they're slightly different levels. Yeah. Okay. So now when we look at these spectra, oh, do you have a question?
So just one more.
Yeah.
So you're not showing the like vibrational levels of the base layer because those are just super unlikely? Or like you're not showing a likelihood of them going up?
The, which ones?
Like if it were
Oh, of being higher here. So for this molecule, we're holding it at a temperature such that we're in the ground state. But you can imagine, you know, you're leaving your vial sitting on the counter, it's warming up, everything gets sort of amplified, right? So you're changing, you can change the absolute energy if you did that. We wouldn't be able to detect it in our instruments, but. So if you look at the measured absorption spectrum of an extract, what you see is the main peak and very distinct side peaks that are the vibrational harmonics. The smoothness of the curve is associated with the rotational levels of each vibrational level of each electronic state. And it's very distinct. You can see these harmonics are very, very distinct. And you can identify them in vivo absorption spectra. There's other pigments in there, but you can identify these side peaks of chlorophyll absorption. And when you measure the fluorescence, you get the main peak and the side peak. And so when you see this mirror imaging of fluorescence relative to absorption, it's because of the quantization of absorption and emission.
So sorry, going back to the probabilities, this would mean that if we plotted a probability density function, that would look directly proportional to the absorption.
Yes. So when you look at different ways of representing, I've just shown the energy levels, but if you plot it versus the internuclear distances, you find that there's shifts and there's a probability to go from here to here that's much more likely, the overlapping probability of where the electrons are located compared to here. Yeah.
Okay.
So you did it. That was awesome. You guys did really well. Yeah, I'm very proud of that. Okay. So this gets into, I'm kind of pulling into a little bit of what Ken was talking about fluorescence, measuring fluorescence. And there's a couple of ways that you can do it. There's something called an excitation spectrum and something called an emission spectrum. A fluorescence excitation spectrum is where you decide where your compound is going to fluoresce and you monitor the intensity at that wavelength. So for chlorophyll, we might pick 683. Or maybe we would pick the side peak. It doesn't matter as long as it's fluorescing there. So you put your detector, you set it to a narrow band, and you're just measuring the intensity, okay? Then you scan your sample by giving it the range of excitation energy. So first, you give it 300 nanometer light. Okay, so you shine a bunch of 300 nanometer light in there and you measure the intensity of fluorescence. And it turns out it's low. Then you say, I'm gonna give it 400 nanometer light. The same amount of energy, just a different color. So however many watts you want to give it the same number of watts and see what the fluorescence intensity is. And it turns out, because chlorophyll absorbs more of that light, it will emit more light. And so the intensity of your fluorescence will increase, right? So you're essentially marching along from the UV all the way to the red, measuring the intensity of the fluorescence. And it turns out the intensity of the fluorescence out here will be low, high, a little bit low, high, really high. It's going to decrease. You can shine 500 nanometer light. You're going to get no signal because the molecule isn't absorbing any light, so it can't fluoresce any light. Then you start seeing a little bit more signal, a little bit more signal, a little bit more signal, well, a lot of signal, and then it will go to 0 if you're measuring out here. Okay. So the excitation spectrum looks like the absorption spectrum because photons absorbed result in photons fluoresced. And if you had a molecule that was in solution, you could quantify that excitation per milligram of chlorophyll, right? You could quantify it. This is what we're talking about with Sedum. We would love to be able to do this with Sedum. The problem is when you do these excitation spectra, your lamp doesn't put out an equal amount of energy at every wavelength. So you have to find a correction factor for your lamp. And so you have to ensure that you know how much energy your lamp puts out in each wavelength so that you get this nice excitation. Or you measure the absorption and you match it so that your excitation matches your absorption, and that tells you what your lamp energy is, and that's actually how you get a correction factor if you put in a known substance, because it's an equal conversion. Fluorescence doesn't care. It doesn't care if it's this photon or this photon. It doesn't matter, remember, because it brings everything down to the base level of the excitation. So that's actually quite helpful. So that's an excitation spectrum. So I'll get to that in a second. Okay, an emission spectrum is measuring the spectral response of the emission. That is measuring this curve. What is the shape? What is the spectral output of fluorescence? So the way that you measure that is you excite fluorescence at one wavelength. And maybe for this case, we're going to pick, because this is an extract in methanol, we're going to pick 420 because that's the peak. And then we're going to scan the emitted photons as a function of wavelength, and we're going to recover this curve. So we're going to give it a certain amount of energy, and we're going to see how much energy it fluoresces at each wavelength. That's the emission spectrum. And you know how much energy you're putting in, but what you want to do is make sure that your detector is sensitive to energy at each wavelength so that you can look at the relative magnitude of that spectrum. And so that's the uniform detection response across your emission wavelengths. These correction factors are not easy to generate for your instrument, which is why people don't tend to do it, which is why you always see things in relative terms, but it can be done. So what do we do with eco-sensors? With eco-sensors, we have one excitation and one emission. So basically, you're just taking, say, the excitation at, well, in this case, we might pick 420, and measuring the emission at 680. Okay? And then we just see what is it here, and what is it here, and what is it in this water, and what is it at this time of day. And we're just measuring the intensity of the response. Okay? But you still need to know these things, even though you're only at one wavelength, and that's why we calibrate. Okay. Questions on that? So I have an instrument that does this. And if you want to try it, we can do it. It was going to be part of the lab today, but I'm trying to not give you too much. But know that we have it. Yeah.
The one part that I'm a little bit confused about is the correction. So you were saying by comparing the fluorescence and the absorption, you can yield a lamp intensity?
Yes. So if you know the absorption of your solution, and it's a solution, it can't be living phytoplankton, right? So if you know the absorption of your solution and you have a molar absorption coefficient, 1 milligram of chlorophyll per liter, doesn't matter where you are in the world, its absorption coefficient will be constant, right? So, and it will have a shape to it, right? It's a spectrum. That will be constant. And so if you measure the excitation spectrum and you get something that looks like this, but then maybe over here it's low, it's because your lamp didn't put out as much energy in the red as it put out in the blue. So if you take the ratio of your uncorrected excitation to the absorption measurement, you'll come up with the lamp energy spectrum.
Okay, I understand. Thank you.
Yeah. Okay. So here's some examples of chlorophyll excitation and emission. Here's phycoerythrin. What's phycoerythrin?
Pigment.
It's a pigment. Who has it? Yeah, cyanobacteria have it. It's a phycobilly pigment. It's a slightly different pigment. I mean, evolved differently. It evolved among the prokaryotes. And so it has a really interesting absorption spectrum. You can see there's, in fact, I don't know whether these are 2 electronic states or vibrational states. I don't know. I don't know if someone else knows. But there's a single emission, and it also has this sort of side peak to it as well. And it's emission, and it emits in the orange. And so it's a really unique fingerprint for cyanobacteria. And here's an example of Sedum. So here is the absorption spectrum of Sedum, and here's an emission spectrum that someone published. I actually just covered it up with a green line so we could see it. But this feature here is actually not fluorescence by Sedum, it's water ramen that they didn't correct for. So you could excite at, say, I don't know where they excited. I think they excited somewhere out in 200 or so. We can probably figure it out. What's the shift to 300?
To 300?
If you got water around at 300, you probably excited at 200?
No, probably at
250?
275.
Oh, excellent. You excite at 275, you'll get a Raman emission at about 300. But then you'll get a big Sedum emission, a big giant peak of Sedum emission somewhere out here, centered on like 450. And so the eco sensors, they excite at 370, which is here, which isn't very high in the absorption spectrum, but it doesn't matter because the emission is going to be very high. And they actually detect around 460, so just off of the peak. So speaking of Sedum, these are the variety of compounds of organic matter dissolved in the ocean or fresh water. I don't know what they are. They have a lot of rings and double bonds, which is why they absorb so strongly and why they fluoresce. The red line is their excitation, their basically absorption, and the dotted line is their emission. And so these are really concentrated on UV absorbing and near blue emission. If we're looking out here at 460, very few of them are actually emitting out at 460. But you can imagine you have a soup of organic matter that's a combination of all of these and others, and you end up with this very broad emission spectrum when you mix them all together. And so it's very difficult to backtrack to try to figure out which of these individual compounds are in the water. But that's what people are working on, trying to use excitation emission spectra to figure out which molecules are in the water. What's the aromaticity? How many double bonds we're talking about? So yeah, okay, very complicated. That's the molar absorption. And one of the things about having a combination of molecules in there in a mixture is that instead of exciting at a range of wavelengths and always getting the chlorophyll fluorescence, always the same shape, always in the same location, when you do that with Sedum, you get the longer you excite, the longer wavelength you excite, the longer your emission shifts, because you're essentially exciting new molecules. that are fluorescing at longer wavelengths. And so people are trying to exploit that to try to figure out what's in the water, but it's a complicated problem. Okay, one of the things about Sedum that you have to consider when you're making a measurement, even with just a little eco sensor, is that the fluorescence by dissolved matter is very sensitive to temperature. Temperature actually really impacts the vibrational levels of the molecule to a very large degree. And I don't know whether it's because it's not in a living organism, but you have to account for the temperature of the water in which the Sedum is dissolved, because it changes the efficiency of its fluorescence. And so here's an example of some measurements being made over a long time. This is November, so it's a year. So over a year, this is the discharge of the river. This is from Brian Downing and Brian Bergamasky's group at USGS in Sacramento. Then they measured the temperature of the water, summertime, wintertime. They also measured the turbidity of the water, because just like you found with scattering, you have to correct for turbidity because it's impacting the amount of signal that gets to your Sedum, and it's impacting the amount of signal of the fluorescence that comes back to the detector. And so they show the difference between just their raw Sedum fluorescence signature after they did the temperature correction, after they corrected for, I can't remember what the color is, but then also they corrected for the turbidity. And so what you see is at two different temperatures, the raw and the corrected are quite different. But when it's warm, they're quite close. It's a very small correction. Okay. When you're looking at turbidity, here's low turbidity water, here's high turbidity water, almost the same temperature. But you can see that there's a very big correction for the path length. So you just, if you're trying to interpret your Sedum, just make sure you take into account the environmental characteristics.
So it's actually impacting absorption, or it's changing the.
And I think it's actually impacting the attenuation along the path of the excitation and the emission. Okay, so that's about all I'm going to say about Sedum. In case anybody has any, we're going to jump into phytoplankton. Okay, proxies. In vitro, in vivo, and in situ. All right, so chlorophyll fluorescence is a proxy for chlorophyll concentration, biomass, and photosynthesis. That's so funny because this is a scalar, this is a rate. Oh no, this is a scalar, this is a different scalar, and this is a rate. But we can do everything with fluorescence. Okay, phycoerythrin, similarly, concentration, cyanobacteria biomass. Oh yeah, Sedum. You can have Sedum fluorescence be a proxy for Sedum absorption, which is quite helpful. Or people use it as a concentration for dissolved organic carbon concentration in some areas where there's a good relationship. Not all carbon absorbs, not all carbon that absorbs fluoresces. So this is a tough proxy and is usually very localized in its robustness of the relationship. All right. Here's the fluorescence signal. When you measure the fluorescence by chlorophyll at its wavelength, which is going to be in the red, it's equal to the energy at the wavelength at which it's absorbed times its absorption coefficient, and we're talking about chlorophyll, so it's the chlorophyll absorption coefficient, times a quantum yield. So if this is the available light, it has units of quanta per meter squared per second, or you could have watts, but we're looking at, we're going to be looking at a specific wavelength. The chlorophyll absorption coefficient, we're going to assume for an extract is constant. We're not going to be able to say that when it's in a cell. It has units of per meter. And then we have this efficiency. This is the quanta fluoresced at one wavelength given the quanta absorbed at the excitation wavelength. So this is a highly spectral term. And so the units on fluorescence, oh yeah, everybody uses phi. It's not the radiant power, it's efficiency. Sorry. We have micromoles of quanta per meter squared per second. The absorption coefficient is per meter. And we have the quanta fluoresced over quanta absorbed, which is dimensionless, except for there's a wavelength shift. And what we get is the quanta fluoresced per cubic meter per second. So it's an amount and a rate. Okay. So if we held the concentration of chlorophyll constant, which meant holding the absorption constant, and we said that the quantum yield is constant because why it always is, right? We could see that looking at this equation, as you increase the intensity of light, you will increase the intensity of fluorescence. Okay, great. If you hold the light constant, as you increase the concentration of chlorophyll via its absorption coefficient, you will increase the fluorescence. So this is easy, right? Looks good. How bad can it be? This is just to show that the fluorescence is nearly isotropic. We talked about why that is. It doesn't remember from whence it came, the excitation. Okay. So in a fluorometer, we can we have chlorophyll molecules in solution, which we see. We have a constant mass specific absorption coefficient. You measure it once, you've measured it because it's in extract and the absorption is not going anywhere, right? Every molecule has the same absorption efficiency, the same absorption capacity. The unit of absorption divided by chlorophyll gives you a very intuitive unit of meters squared per milligram. It's absorption per meter divided by chlorophyll milligrams per meter cubed. When you do the units, the meters cubed cancels. Okay, that's constant. Because there's no physiology in a test tube, the quantum yield actually is constant. And if we are good at our fluorometry, we can maintain a constant irradiance. So if this is constant, and this is constant, and this is constant, then what does it say about the fluorescence that we measure? It's proportional to chlorophyll. Okay. So we measure the fluorescence in relative units, and we have to calibrate our fluorometer. But what we end up with is a chlorophyll concentration. This is the basis for extractive chlorophyll using a Turner fluorometer. You will do this today. And you will be able to calculate the chlorophyll concentration of an extract. And the assumptions are minimal. Okay.
So on the graph where there's 0 extracted chlorophyll and there's a signal, is that from the slope?
That's the instrument offset.
Oh, okay.
Yeah. So slope and intercept. So you do this calibration on your instrument, on your fluorometer, with a known concentration that you bought from a chemical company.
Okay.
Yeah.
And they tell you what the fluorescence.
They tell you the concentration of chlorophyll. You measure a dilution series and a spectrophotometer, so you know the concentration based upon a molar absorption. And then you look at your fluorescence response of your benchtop fluorometer. That's all. You have to calibrate everything. Yeah. Okay. So we'll do this in the lab. All right. Now, in vivo fluorescence physiology. Phytoplankton contain a multiple chloroplasts, which are the membrane-bound organelles. So here's a beautiful diddleum, and you can see each one of these chloroplasts. This is from the IFCB. Inside each chloroplast, you have these stacked structures called grana. Each one of them is a stack of thylakoid membranes, which is the two-dimensional hydrophobic structures here. And embedded in that is this pigment protein complex. We call it a light harvesting complex. It's a photosystem. So that's where the pigments are. And that is the site of photosynthesis. The chlorophyll molecules, I already told you, have a phytol tail. That is structural. That's actually embeds them right into the membrane and keeps them oriented so that then the absorbing faces are facing outward, available to collect light. Okay. The chlorophyll molecules in this light harvesting complex are responsible for absorbing blue light, the 440s. When they are in this complex here, we call them antenna chlorophyll because they are out there collecting radiation. There are other pigments out there. In this example, they show you an orange one to be the carotenoid, which absorbs more orange light. Also, chlorophylls B and C are out there, and they're absorbing a little bit longer wavelength light. So we're starting to absorb into longer wavelengths, shorter energy in the cell. There's a process by which any one of these molecules can absorb light within their absorption spectra. And they start passing that energy around between each other. But ultimately, they're funneling that energy into the reaction center for photosynthesis. The transfer of energy between them is, if they have overlapping absorption spectra, is lossless because they can have overlapping electronic states. And so they call it radiationless and lossless dipole-dipole resonance. I mean, literally the molecules are next to each other, they're in an excited state, and the energy is passing between them. Ultimately, that energy is transferred to some really specific chlorophylls in the reaction center. And those chlorophylls are bound together. There's like two of them bound together into a dimer. One's bound with a phaophyton in a dimer, depending upon which of the two photosystems you're talking about. We call them P680 and P700. That's the photosystem 2, which is actually starts at the beginning, is P680 because it absorbs at 680 nanometers. This reaction center absorbs at 700, slightly longer, slightly less energy. All the energy that was absorbed in the blue and the green that isn't 680 or 700 has been lost to heat. Once those reaction centers are excited and they're at their electronic, excited electronic state, rather than decaying back to the ground state, they just pass off their electron, right? Which takes them out of the excited state, but also leaves them without an electron, right? So in photosystem 2, that's where you split water and replace your electron. If you're in photosystem 1 and you've gotten rid of your electrons, you're getting basically an electron from photosystem 2. So water gets split, oxygen is made, we're passing electrons along the scheme, but really what we're doing is physically in the cell is we're moving electrons through these thylakoid membranes and setting up proton gradients. The process results in, so we split water, but the process results in the formation of the reductate and ADPH, as you remember, also the capacity to make ATP, both of which are used to fix carbon. So my question is, what does this have to do with fluorescence? And really, why are phytoplankton fluorescing if they need all that energy to go through that whole scheme to fix carbon? So it's the chlorophyll molecules in the light harvesting complexes that do most of the fluorescence. And what happens is when so much energy is being absorbed by the light harvesting complex, and I'm passing to you, I'm getting energy, I'm passing, and suddenly you can't take any because you've already absorbed your own energy, there's a backup. And the energy is getting into the reaction center and it's operating as fast as it can. There's just inefficiency. And so what do? You fluoresce, just get rid of it. That's a lot of energy. You don't want that much energy being released as heat, so you release it as a photon. So if we look at the environment for phytoplankton, initially, the more light that is absorbed, so if you're in a low level of light, you absorb light, you do photosynthesis, and you're sloppy, so you have some fluorescence. You absorb a little bit more light, the intensity is going up, it's mid-morning. Great, do a little bit more photosynthesis, still sloppy, a little bit more fluorescence. So initially, the more, when you start off with low light and you increase it a little bit in this light limited regime of the phytoplankton, the more light you give them, the more they photosynthesize and the more they fluoresce. So fluorescence and photosynthesis co-vary. So fluorescence becomes a good proxy for photosynthesis. So that's this part of the scheme. And this is actually for eelgrass, but it was the best I could find. But initially, this is photosynthesis and this is fluorescence. And initially, it's a linear, you couldn't distinguish this from linear. They're related. But as photosynthesis starts to get to its maximal rate, starting to get into the saturated regime of the photosynthesis or radiance part of the curve, photosynthesis actually starts to saturate, but they're still absorbing a lot of photons. So the excess photons go into excess fluorescence. So fluorescence goes up. That's this part of the curve. Fluorescence goes up for an equivalent increase in photosynthesis. So suddenly now they're not so covariate. You have enhanced fluorescence relative to photosynthesis. And that's when you're starting to get some photoinhibition of photosynthesis. Eventually, if you keep giving them light and they keep absorbing it because pigments absorb light, now you reach a regime where not only have you inhibited photosynthesis, but now you inhibit photofluorescence. And that's where you get into this non-photochemical quenching. So now both your photosynthesis and your fluorescence are damped. And we typically see this happening at noontime in the environment. So yeah, fluorescence can be a good proxy for photosynthesis here and here, but maybe not here. And how do you know where you are? Okay. So to first order, fluorescence is the proxy for chlorophyll. You have more chlorophyll in the water, you're getting more fluorescence. Under some light conditions, limiting conditions, fluorescence is a good proxy for photosynthesis and the more chlorophyll you have, the more absorption you have, the more photosynthesis you have, the more fluorescence you'll have. But then you get this place where photosynthesis starts to saturate before fluorescence does. And so then it's not such a great proxy. And then under saturating levels, both of them drop off. So it maybe becomes a good proxy, but maybe not for the best reasons. Okay, where are we time-wise? Yeah.
Is the light intensity that you shine for things, like if you have another sensor that measures the intensity of the light, can you know what point you are on that?
So if you are out in the environment and you have a radiometer and you're keeping track of light, you definitely know where you are. And you can definitely see quenching of fluorescence. And I can show you that in the mooring that we have out there. It's part of what at the end we can see something about that we won't get to today. What's the time?
11:30.
Oh. Everybody stand up and take a big breath because it is hot in here. And I'm hot. And if I'm hot, you guys are hot. Whew. Yeah, so it's really nice having this mooring out here because every hour it reports in real time the intensity of light and the intensity of fluorescence. And you see that as the sunshine goes up like this, the fluorescence does this.
But actually decreases. It's not just that the linear relationship stops.
Fluorescence gets quenched. Yes.
So what's happening to the light energy? Like,
It's heading off as heat. It's heading off into these weird triplet states of energy dissipation. And honestly, it can just go into breaking bonds.
does it degrade?
Yes.
Okay.
And so I always think to myself, why would this cell design itself so that it's going to quench every day at noon and sometimes, structurally damage the cells? And I don't have an answer for that. I guess it's not that expensive to fix things. And they get enough photosynthesis in the morning and the afternoon that they take a break at lunchtime. I don't know. Yeah.
What happens on a cloudy day?
It doesn't go down as much. You can
Does it go
track it
down?
to clouds moving over.
Does it go down at all?
Yes, it depends on how thick the clouds are. It's literally proportional to the intensity, and you can model it, and I do. But yeah, you can use fluorescence as a tracker of irradiance and a tracker of whether you have uniform clouds or patchy clouds. So there's a very immediate response, there's a recovery time, and then there's sort of that, oh, I have to make my molecules again type of recovery time. Okay, back to this equation. So we did this for in vitro in an extract. Now we're doing it in the cell. So the molecules of chlorophyll are bound to proteins and embedded in membranes. So what that means is when the molecule absorbs light, that energy has other places to go. It can go to photosynthesis. It can go to heat dissipation. It can go to these sort of weird pigment energy degradation or energy dissipation pathways. So the variation in the quantum yield, it varies a lot. So the quantum yield in a living cell is about 1 to 3%. But if you're doing the modeling, that's a factor of 3. Just that. And in fact, it's less than 1%. So that could be a factor of 6. Hold that number. 6. I mean, you can vary the estimation of chlorophyll from fluorescence by a factor of 6 just because of the quantum yield, because there are other energy pathways. Also, we know that phytoplankton absorption per chlorophyll varies. We talked about how if you have a big cell with not very much pigment, because they're in highlight, bless you, and a big cell with a lot of pigment, the absorption for chlorophyll will be really different. If you have a big cell and a small cell, because of the package effect, the absorption for chlorophyll will be really different. Huge factors. So we have a ton of variability here and a ton of variability here. That's why they're red in this equation, because those are very large sources of variability. So we still have our great fluorometer, so we still can maintain a pretty constant excitation, but that's the only thing that's constant. This is variable and this is variable, and we're trying to estimate this from fluorescence. And again, fluorometer is relative units. So we still have to calibrate our fluorometer. So we can calibrate this measurement versus this measurement from the slope and intercept. But depending upon how we calibrate it, we are pre-selecting our specific absorption coefficients by our calibration standard. And if it's a living cell, we better know something about where this term is. How bad can it be? Oh, it must be a straight line. It's going to be great, right? We're doing this today. So you guys will be divided into 4 groups. One group will get Denaliella. One group will get Thalassia syra. One group will get Harpswell sound water. And you guys are going to keep everything really dark because we don't want it getting inhibited. But one of you is going to get Harpswell sound water in clear bottles. And you guys are each going to calibrate the same fluorometer with these 4 different samples. And then we're going to compare the calibrations. And those calibrations are going to tell you something about that. We're just going to see how bad can it be? How bad can it be? All right. How many of you experienced this? You just got your fluorometer back from the factory. They gave you a calibration. It's written on a nice piece of paper. Sometimes that paper is laminated, so it looks really official, and you can put it in your notebook. Or even you calibrate it yourself, and you're like, I'm good at this. I've been doing this for 20 years. Perfect calibration. And then you go into the field, You feel all confident, you make all these measurements, you collect a water sample, you measure the extracted chlorophyll. You're good at that. You've been doing this a long time. You're a good filter. You have very low error in your replication. You plot it up together, expecting a straight line, and you get this. So Scott, this is Scott Libby. He works for Battelle. I've known him for since graduate school. He's smart. He's capable. They get this data. This is from Massachusetts Bay, 1,000 paired samples. They go out every month. They visit all these stations. They do their fluorometer. They lower it through the water. They collect water samples. They start at 7. They finish at 4. They do the samples in the same order every time. Plot them all up. And it's just a nightmare. There's just no relationship between what the instrument tells you and what your extracted samples are. So what's the problem here?
We had those 2 red terms that had potential variability. So you can imagine that there's a lot of different types of phytoplankton there.
Different phytoplankton.
Differently, and then throughout the day, you could have different quantum yields.
Different quantum yield because of quenching. You're going as a function of depth, quenching in the surface, not quenching at depth. Phytoplankton, different species, live differently in the water column. You always go to station 1 at 7 in the morning. You always go to station 10 at 4 in the afternoon. What time do you do the noontime sample? It's always station 3. Station 3 always has low chlorophyll. No, station 3 always has low fluorescence because it's at noon, right? So they actually gave me this data set 15 years ago and said, we don't know what to do. So I went through and I figured out all the metadata and I figured out, and you could actually plot it. It was like, oh, station 1, station 2, station 3, station 1. Like you could plot it by time of day and depth, and the fact that, you know, they do everything in a certain order because it's a contracted business. So you can reduce the variability here significantly if you think about it. Okay. So here's what we're going to do. We're going to go and do a quick overview of calibration. We measure volts. We want milligrams per meter cubed. Standard. What are we going to use? How would you calibrate something to chlorophyll? You want to know what the chlorophyll response is. What should we calibrate to? Chlorophyll. That would seem so incredibly obvious to me. So some people use a solid standard. They give you those. They trace the signal, but they don't convert you to from volts to chlorophyll. They just tell you whether your instrument is not drifting. So don't, when they sell you the solid standard for a lot of money, it's not really what you're looking for. So you would think that you would wipe chlorophyll A and do a dilution series and measure it. The problem is the excitation on the fluorometers is what? Anyone know what the excitation on your chlorophyll thermometer is? 470, 460. It's probably 460, but they tell you 470. So that's here. What's the absorption coefficient of your extracted chlorophyll? 0. Doesn't absorb there. So you can't calibrate with the extract. So you have to use a living cell, because the living cell has a pigment that does absorb there, and in the light harvesting complex, it passes its energy ultimately to chlorophyll, which fluoresces. So that's fine. But you have to think about who you're going to use. Right? So there's nice little chlorophyll, fluoresce. Yeah, your excitation mismatch. Okay. So I'm giving you 4 different things. We'll see how different it is. How different is your calibration depending upon which culture or natural sample you use to calibrate. Also, you can't put the sensors into a solvent or you will completely degrade the optical window on your sensor. Yeah.
And the ratio of pigment to chlorophyll varies from phytospecies. You cannot just measure the pigment.
We'll see. That's what they tell you. Okay, so pick a living culture. So this is an example from an experiment I did with one of my former students where we grew a bunch of cultures and let them get really thick so you can see the color because they're really pretty. This one happens to be the diatom Phyllosera. So maybe if you're going to be doing something you're going to trace through time, pick one that's easy to culture. Pick one that's ubiquitous to the environment, that has robust optical properties. So if I measure it this year and next year and the year after that under the same growth conditions, I get this in calibration. I found Fluorescence pseudonana was a pretty good one to work with. So I keep its growth conditions with plenty of nutrients and non-inhibiting light. I put it in a 24-hour light cycle so that it doesn't undergo synchronous division. It's sort of, if you grow it on a light dark cycle, they're all growing and dividing, and then their cellular chlorophyll is different. But if you keep that from happening on average, you get like an average scenario. And I capture them in exponential growth when they're not dying. Sadly, yours are sort of sad in the lab, but we'll pretend they're an exponential growth. And then I just maintain a database of the chlorophyll concentration, the other pigments, the absorption, its size, and its carbon, just to make sure that I'm keeping this culture. Okay. So I create a dilution series. You guys are going to do a dilution series. And then you measure the fluorescence of each dilution. You also do the fluorescence of whatever you're diluting with, which is your filtered seawater. And then it's best if you do this in a dark casket. But these are 4 liters. We only have one of them. They're all in England. So you're not going to use the casket, but when you do this at home, tell your colleagues, do it in the dark vessel. We're going to do it in a beaker on a black cloth in a dark room. It's actually not as bad as you think because the path length is very short. And then from each of these, you're going to extract the chlorophyll. You'll take 3 samples, measure it in the Turner fluorometer, and get the concentration of chlorophyll from the extraction where it works because you don't have physiology. And then you can put numbers on here. And you'll have error bars because you're going to have triplicates of your chlorophyll. You're going to have a minute of fluorescence measurements so you can get uncertainty in both directions. And you're going to calculate a linear regression. And if you use a type 2 regression, it takes into account the standard deviation. So that was a really big standard deviation. So you might not want to weight that point as much as say like this one, which is really tight standard deviation. So that's the type 2 helps you out with. And you get a calibration slope, which converts the fluorescence to chlorophyll. That is your fluorescence yield for your calibration. That is the fluorescence per chlorophyll for your culture in its physiological state. That's all the calibration slope. The intercept is your blank signal. It's the medium blank. It's not the dark. The dark you actually have to measure with tape again, right? Because what you're looking for is an equation to estimate chlorophyll from measured fluorescence. So what you want is to take a measurement of chlorophyll in the ocean, subtract the dark, and then divide by the slope in order to estimate chlorophyll. So that's what you're going to do. Okay, so where am I going with this? Oh, yeah. Okay. For your particular sensor, I don't want to talk about this right now. The point is, you want to be able to do this so that you can compare your measurements, what you do today with what you do tomorrow. You want to compare it with your other sensors, and you want to compare it to your colleagues. And ultimately, we want to be able to create climate quality data records of chlorophyll concentration or whatever it is you're measuring. So you need to be able to have your calibration be robust for 4 decades. So whatever you do, you need to be able to do the same thing in 4 decades so that you can recover a comparable estimate of chlorophyll using your method. So I don't know what the best way is. I'm just going to teach you one way how to do it. And if you improve on it, please tell me, because that would be nice for me. Okay. So given sources of variability, what can we learn? Well, we know that it's lunchtime. So there is so much more that I can say. I could talk for another 2 days on fluorescence. Simple to measure, difficult to interpret. I have a fluorometer that has multiple excitations and measures chlorophyll. If you're exciting in different parts of the spectrum, you're exciting different pigments. So this instrument begins to tell you something about the community composition. And it's beautiful. And I would love to spend some time telling you about that. I also have a lot of experience putting fluorometers on different platforms. There are very different considerations depending upon if you're profiling from boat, if you have a moored sensor. I can talk about biofouling until I'm blue in the face. Profiling on gliders and floats. There's a lot to be said about non-photochemical quenching and how you correct for it so that you can retrieve chlorophyll concentration at noon. So we can talk about this stuff in the next few weeks as people are interested, but we're not going to do it now. Yes.
I have a nomenclature question.
Yes.
So when you have more intense light at noon, you get photochemical quenching, but that to me sounds like photochemical quenching.
So photochemical quenching is when the fluorescence is lower than it might be because the energy is going to photosynthesis. So photochemical quenching is actually what phytoplankton want to do. They quench their fluorescence in favor of photosynthesis. They're channeling energy efficiently into photosynthesis. So fluorescence is lower than if you block the photosynthetic pathway, your fluorescence would go way up. Okay. I know. It's the who's doing the analysis? You photosynthesizers, you're quenching my fluorescence. Oh, you fluorometer, you're quenching my photosynthesis, right? Like who's quenching whom? Yeah, it is. Takes you a lot to think about it. Any other questions? Yeah.
How fast does it change in, like how fast is quenching the or in?
When the sun comes up, you start to see it. It's
Let's say
so
we're in
10
lab
o'clock.
and I
So
accidentally shine it
oh,
and
so
go back.
so one of the things that you have to do when you're doing your fluorescence measurement, you have to move your fluorometer. If you take your fluorometer and you put it in your sample and you hold it there, you'll watch the fluorescence just go down. You will quench it with your fluorometer. So you just slowly move around. You can't tilt it because otherwise you're going into the sidewall. So you're just going to slowly move it around to keep that from happening.
If I like walk in front of a window with this sample, 10 minutes later, will it
Oh.
be responding to that?
To like a quick move
To
by?
a
No. So the longer you expose it, the longer it takes to recover. The higher the light you expose it to, the longer it takes to recover and the more that it quenches. So you can quench at noon in stable water column. You can quench your fluorescence by 90%. So you would say, oh, my chlorophyll went down from 10 to 1. It's crazy.
So you're saying if that cloud goes over, you can see that.
It goes down a little bit.
couple minutes.
Yeah, it goes down and then it will come back up. Yeah. Or it goes up when the cloud comes by and then it goes back down. Yeah. So what's really troublesome out here is because you have such huge tidal variability and it's out of phase with the diel cycle. So I had a student that we just published a paper on where he was able to deconvolve the tides from the sun. And in doing that, you can correct for non-photochemical quenching. You can recover the original fluorescence of the high tide and low tide water and track the populations in the two water masses through time. It is really beautiful. And I've run this model on places that have relatively low tidal variability, but as long as you have some tidal variability, it's very cool. And the reason why we figured it out is we expected our fluorescence to come up at night. And it would come up at night and then it would dip down. And then you realize like, oh, it's because low tide was at midnight and it brought a new population in. And so it looked like quenching, but it wasn't, it was just a different population. And so when the population decrease is of the same order as the quenching decrease, it was really complicated, but it was a really fun little project that a senior could do. So there's a lot of nice fun games you can do with it. Yeah. We good?
I have a random question.
I'll have a random answer.
For the chlorophyll A absorption,
Yeah.
At 500
Yep.
grain, there's like no absorption, so it's not completely elastic.
So that is in an extract. that just has chlorophyll molecules in a solvent. And it looks green to you because it's absorbed the blue and the red light. And whatever is in your solution has some scattering, and the light that is left to be scattered back into your eye is the green. Yeah. Physics for babies. Is that picture in there? I was like, it's green because it absorbs. Yeah.
When you have quenching from noon and you wait, maybe it's 2 o'clock.
Or 4.
Or 4, there's still quenching, and you want to recover that. This might be pushing the envelope of what we want to discuss before lunch, but how do you recover that?
There are a lot of strategies that people use. So if you had constant chlorophyll, just imagine there was constant chlorophyll through time, and over the day you would see this. You could just draw a line. You could just average from midnight to midnight, and you could estimate it, and the shape of it would probably look the opposite of your light. So cool, except that you don't have constant chlorophyll through time, because if you did, why would you have a mooring out there? Right? So then it's like, oh, well, I've got stuff is increasing, so I can draw the straight line. And that's exactly what we were doing until we figured out that we were getting really low values at a low tide at night, and so we couldn't draw across our midnights because it was a different population, which is why we did the project to begin with. So you can do that, you can draw that across, but you are making a lot of assumptions that you've had no change in phytoplankton community from midnight to midnight. Some environments that might work great. So in profiles or with profiling floats, when you have quenching, so you have maybe a chlorophyll that looks like this, but then your fluorescence does this, right? And you have this subsurface chlorophyll max, but you don't. You have a subsurface fluorescence max. There's still phytoplankton in the surface. So what people do is they sort of get to their maximum, and as long as you're in the mixed layer, they just project that maximum value at the base of the mixed layer to the surface. And just say, it's a mixed zone. I'm sure the phytoplankton are uniformly distributed. So that's solution 2. Other people look, oh, yes. Other people look at the profile of backscattering or beam attenuation to tell something about the distribution of particles. So they come up with a ratio of backscattering to chlorophyll in a region of the water column where there's no quenching, and then use the backscattering profile to, and that constant ratio. Backscattering to chlorophyll ratio, it depends on, it will vary, but is that your biggest source of error? Maybe not in that situation. But after spending a month on the exports cruise where we did 100 profiles and people continued to collect samples at the chlorophyll max, and I was like, it's not. I've measured the absorption, it's uniform. The top 30 meters are uniform. The chlorophyll is constant, it's high at the surface. It's just the fluorescence is low. There was no subsurface max. It's just the fluorescence max because it went away at night.
So what are people actually sampling then when they sample the subsurface chlorophyll max?
In this case, they're sampling the population of phytoplankton at the base of the mixed layer. And hopefully they're also sampling the surface, and they're finding they're the same.
When I extract the
Yes, I measured the absorption, and it was the same spectrum. It was the same community. And there must have been enough mixing to keep them from acclimating to the light from the top to the bottom of the mixed layer. Yeah. We also measured the IFCB on those samples, and they were the same. Yeah.
So chlorophyll maxes are
Some,
always just fluorescence maxes?
nope. Some, when you measure fluorescence profiles and you're doing it at noon, if you have the subsurface fluorescence maximum, it's probably because you're quenching at the surface. You don't, it doesn't mean that there aren't cases where there are fluorescent deep chlorophyll max, but just make sure you've measured it with something other than fluorescence at noon, just to verify. Yeah. Cool. Let's go eat. Oh. Where? Oh, at what time? Well, it depends on the light. If it's a clear day, you get the minimum right around when the solar and the sun is at maximum. And it seems to recover pretty uniformly, right? As long as if the cells are held really superficially and it's hot and it's sunny and they have all sorts of stresses, it can take them a little bit longer to recover at the end of the day. But the response to turning the lights down is very quick. As long as they're not nutrient limited, they haven't been somewhat denatured. There's some really interesting work in the Antarctic by John Cullen and Pat Neal and Richard Davis, where they looked at fluorescence and photosynthesis of Antarctic phytoplankton. And there was so much UV light because of the ozone hole. There was incredible quenching of fluorescence and photosynthesis. But if they could get mixed down, there was enough mixing so that they could get a chance to recover, they could recover their photosynthesis. But if the mixing rate was slow, they just got fried and bonds got broken and there was no recovery. And he called it the conveyor belt of doom. So if it was a fast conveyor belt, you were fine. But if it was a slow conveyor belt, the cells would just get fried. So first would be inhibition, then would be quenching, then would be damage, depending on how long, yeah.
So is the loss of fluorescence by quenching, by highlight, lower than you would see with like block fluorescence just from like lack of light and lack of photosynthesis? So very vectors.
To first order, your fluorescence is proportional to chlorophyll. To second order, your fluorescence is proportional to whether you're in low light or high light, right? So if you have a factor of 10 difference in chlorophyll, you'll see a factor of 10 difference in fluorescence. But you can also see a factor of 10 difference in fluorescence between quenched and unquenched for the same concentration. Yeah, it's hard to interpret what it is. Yeah. Yeah. All right, cool. So it's going to take me just a little bit to get things set up down in the lab. So why don't you guys come down at 1:30 just so I can make sure that the lab has been transformed from yesterday. It's mostly ready. Yes.
But if there's zero absorption and extracted pigments at 465 or 70, why are they fluorescing wrong? But the fluorometer only sends a very
Yeah, they won't fluoresce. They won't fluoresce because there's no excitation.
Isn't that what you're measuring?
So if it's an extract, you have a fluorometer that has an excitation at 435.
Oh, okay. We just use the same fluorometer. So
Yeah.
ours must be regularly.
Yes. So our Turner fluorometer has an excitation at 430.
Okay, I think
And
we have almost.
and so that will excite the extract. It can also excite the in vivo, right?
Right.
But